Good morning everybody. Today I am going to discuss metabolism of basic amino acids. Histidine, arginine, lysine are the three basic amino acids. Histidine and arginine are essential amino acids and they are required during pregnancy, lactation and should be supplied through diet. Both histidine and arginine are glucogenic amino acid, whereas lysine is predominantly a ketogenic amino acid. Coming to metabolism of histidine in detail, our metabolic role or functions of histidine includes: histidine plays an important role in protein biosynthesis. It is also necessary for synthesis of glucose. This histidine synthesizes biologically important dipeptides, anserine and carnosine. Histidine also converts to biologically important amine, histamine, by decarboxylation reaction. And also, histidine necessary for formation of glutamate and for amino tetrahydrofolate. Coming to catabolism of histidine, histidine is glucogenic amino acid and it is catabolized to glucogenic end product alpha-keto glutarate, which in turn converts to glucose through gluconeogenisis, or histidine may enter into TCA cycle for oxidation. Histidine undergoes deamination with the help of an enzyme histidase to form urocanate. This urocanate formed from histidine converts to 4-imidazole propionate by adding a molecule of water with the help of an enzyme Urocanase. Imidazole propionate converts to form aminoglutamate or figlu by adding a molecule of water with the help of an enzyme 4-imidazole propionate hydrolase. This form-aminoglutamate or figlu converts to glutamate with the help of an enzyme glutamate form-aminotransferase, which requires tetrahydrofolate as coenzyme. The glutamate undergoes transamination with the help of an enzyme transaminase to form alpha-keto glutarate, that is the glucogenic end product of histidine. So alpha-keto glutarate finally converts to glucose through gluconeogenisis, or it enters into TCA cycle for oxidation. In folate deficiency, conversion of figlu to glutamate cannot be carried out because it requires coenzyme tetrahydrofolate. As a result, figlu accumulates in the blood and excreted through urine. Increased figlu excretion in urine indicates folate deficiency, and this folate deficiency is detected by histidine load test that we will discuss little later. One more metabolic function of histidine is formation of histamine, already discussed under biologically important amines. Histidine undergoes decarboxylation to form histamine. This histamine is formed in basophils, mast cells, and to some extent in gastric mucosal cells also. Coming to functions of histamine, this histamine acts through types of receptors, namely H1, H2 and H3 receptors. H1 receptors are present in platelets, mast cells, basophils, and they act through cyclic GMP. Cyclic GMP acts as a second messenger. H1 receptors are responsible for vasodilatation, thereby increases BP, causes bronchospasm, and also involved in mediation of allergic reactions. H2 receptors are present in gastric mucosal cells and they are responsible for gastric acid secretion or gastric hydrochloric acid, gastric HCl. Coming to H3 receptors are present in presynaptic nerve endings and are responsible for synthesis and release of histamine in brain and these S3 receptors acts as CNS stimulant. So overall functions of histamine are: histamine acts as anaphylactic and anti-inflammatory agent. Histamine causes contraction of smooth muscles. Histamine acts as vasodilator. Histamine increases the gastric acid secretion, that is gastric HCl. Histamine also acts as neurotransmitter and it causes pruritus. Coming to antihistamines. Antihistamines are the compounds or drugs having structural similarity to histamine and they prevent physiological changes produced by histamine. Chlorpheniramine and diphenhydramine are H1 receptor antagonists, whereas cimetidine and ranitidine are H2 receptor antagonists and are commonly used drugs for the treatment of peptic ulcer. Coming to one more important function of this histamine. Histamine helps in the synthesis of biologically important dipeptide carnosine and anserine. Anserine is dipeptide consisting of histidine and beta-alanine, whereas anserine is nothing but methylated carnosine. This carnosine is formed from histidine. Histidine reacts with beta-alanine. With the help of an enzyme, carnosine synthetase forms carnosine, which requires a molecule of ATP. This carnosine formed from histidine undergoes methylation with the help of SCAM to form anserine. Carnosine and anserine are biologically important dipeptides. Both activates the activity of an enzyme myosin ATPase and also acts as copper chelating agent. The fourth important function of this histidine is it acts as a donor of one carbon metabolism, that is formate. We will discuss this function under one carbon metabolism. Coming just now I told you histidine load test will be discussed little later. Coming to histidine load test. This histidine load test is used to assess folate deficiency. About 5 grams of histidine is given in 3 or 4 divided doses at an interval of 3 to 4 hours and urine is collected for 24 hours after the initial dose. Normally less than 30 mg of fig glue is excreted in urine, but in case of folate deficiency, fig glue excretion is increased more than normal. Coming to disorders associated with histidine metabolism. Histidinemia, histidinuria and urocanic aciduria are the 3 inborn errors of metabolism associated with histidine. Histidinemia, a rare autosomal recessive disorder, and it is due to deficiency of an enzyme histidase, and it is characterized by mental retardation and speech defect. There is an increased concentration of histidine in blood and increased excretion of histidine in urine. Also, there is an increased excretion of imidazole, pyruvic acid in urine. These are the 3 important biochemical changes in histidinemia, that is increased concentration of histidine in blood, increased excretion of histamine in urine and also increased excretion of imidazole pyruvic acid in urine. Coming to histidinuria, a rare inherited disorder and it is a benign condition and also a asymptomatic condition due to defect in transport of histidine from renal tubules. Urocanic aciduria is due to deficiency of an enzyme Urocanase. Also, it is also a benign harmless condition and there is an increased excretion of urocanic acid in urine. Coming to metabolism of arginine. Here, instead of asking metabolism of arginine, they may ask some 5 marks question as short essays. That is one of the most common Question in this arginine metabolism is nitric oxide and its importance, polyamines and its importance. They may ask the creatine synthesis also, and also glucogenic or catabolic role of arginine. Arginine is basic, non-essential and glucogenic amino acid. It has an important function in cell division, wound healing, immune function and also involved in urea cycle. It is used in the treatment of erectile dysfunction, dentine hypersensitivity and also used as vasodilator. Coming to catabolic pathway of arginine or functions of arginine. Arginine is used for synthesis of nitric oxide. It is necessary for synthesis of creatine along with glycine. Arginine plays an important role in protein biosynthesis and also arginine plays an important role in urea cycle. Arginine is necessary for synthesis of polyamines, that is putrescine, spermidine and spermidine. Arginine first converts to ornithine and this ornithine acts as precursor for synthesis of polyamines. And also this arginine is a glucogenic amino acid. Coming to synthesis of ornithine from arginine. Ornithine is a non-protein amino acid synthesized from arginine by two ways. Number one, arginine converts to ornithine with the help of an enzyme arginase and arginine converts to ornithine and urea with the help of an enzyme arginase. This occurs only in the liver. Number two, the arginine with the help of an enzyme arginine glycine transaminidase to form ornithine and guanidinoacetic acid. This reaction occurs in kidney and other tissues but not in the liver. Coming to nitric oxide, it is an biologically important compound synthesized from arginine through the formation of ornithine. Nitric oxide is synthesized from arginine. Arginine supplies nitrogen atom for synthesis of nitric oxide. The enzyme nitric oxide synthase converts the enzyme nitric oxide, which is a dimeric protein consisting of heme, FAD and tetrahydrofolate. This nitric oxide synthase dimeric enzyme converts arginine to nitric oxide and citrulline, and it requires calmodulin for its activity. Here, one atom of oxygen is added to nitric oxide and the other atom to citrulline, and hence enzyme is called as dioxygenase. There are three isoforms of nitric oxide synthase. All forms are seen in almost all tissues and these are product of different genes. There are three isoforms: NOS1 or neuronal nitric oxide synthase. It is present in central and peripheral neurons. It is mainly a cytoplasmic enzyme. Its activity is activated by calcium. The gene for neuronal NOS is on chromosome number 12. Coming to NOS2, or it is also called as inducible enzyme or macrophage nitric oxide synthase, are also called as INOS, nitric oxide synthase. It is found in macrophages, neutrophils, hepatocytes. It is induced by cytokines during inflammation. It is also a cytoplasmic enzyme and it is not activated by calcium. The gene for nitric oxide synthase is on chromosome number 17. NOS3 or endothelial NOS. It is seen in endothelial cells platelets, endocardium, myocardium, and it is located in plasma membrane, not in cytoplasm. It is also activated by calcium. The gene for nitric NOS3 is on chromosome number 7. Coming to physiological functions of nitric oxide. This nitric oxide acts as a potent vasodilator and thereby increases BP. Nitric oxide inhibits platelet aggregation. This nitric oxide also causes relaxation of smooth muscles of intestine and urogenital tract. Nitric oxide acts as neurotransmitter. It increases renal excretion of sodium and water. Peroxynitrite formed from nitric oxide causes lipid peroxidation. Coming to therapeutic uses of nitric oxide, glyceryl trinitrite used in the treatment of angina pectoris. This nitric oxide is also used in the treatment of pulmonary hypertension and also used in the treatment of impotence, and also it is used in the treatment of shock. N-monomethyl arginine and asymmetric dimethyl arginine are the two important antagonists of nitric oxide. Coming to polyamines. Polyamines are the substances containing multiple or many amino groups. Putrescine, spermidine and spermine are the three polyamines. Spermine and spermidine are detected in semen, hence they are so named. These polyamines are synthesized from arginine. Coming to synthesis of polyamines from arginine. Arginine, with the help of an enzyme, arginase converts to ornithine. Ornithine undergoes decarboxylation with the help of an enzyme, decarboxylase, to form putrescine. This putrescine reacts with decarboxylated SAM, which is formed from SAM to form spermidine with the help of an enzyme, spermidine synthase. This spermidine converts to spermine with the help of an enzyme, spermine synthase. It also requires one more molecule of decarboxylated SAM. Here, S-adenosylmethionine does not participate as methyl group, but it donates propylamine moiety. Coming to functions of polyamines, these polyamines are involved in cell proliferation and growth. They are involved in synthesis of DNA, RNA and proteins, or plays an important role in transcription and translation. Inhibits the activity of an enzyme, protein kinase. These polyamines are involved in stabilization of cell membranes. Coming to clinical importance of polyamines. Increased polyamine excretion in urine occurs in all types of cancers like leukemia, CA lungs, bladder, kidney, etc. Putrescine is an ideal marker for cell proliferation, whereas spermidine is an ideal marker for cell destruction. This is the clinical importance of polyamines. These polyamine excretion is increased in all type of cancers. Coming to metabolism of lysine, here they are not going to ask the metabolism of glycine. If at all they want to ask lysine metabolism, they are going to ask only carnitine, what is carnitine, how it is synthesized, and what is its role in beta oxidation of fatty acids. I think already we discussed under metabolism of lipids, that is in beta oxidation of fatty acids. Lysine is essential, basic, predominantly ketogenic amino acid. Meat, fish, poultry and dairy products are the rich sources of lysine. Hydroxylysine is an important constituent of collagen and elastin. Trimethylylysine acts as precursor for synthesis of carnitine, which plays an important role in oxidation of fatty acids. Coming to catabolism of lysine. Lysine is ketogenic amino acid and it is catabolized ketogenic end product acetyl-CoA through a series of reactions. Okay, this is the catabolic role of lysine. Lysine converts to saccharopin with the help of alpha-ketoglutarate, undergoes dehydrogenation to form saccharopin. This saccharopin undergoes dehydrogenation to form alpha amino adipate delta semialdehyde with the help of an enzyme dehydrogenase. Again, this semialdehyde undergoes dehydrogenation with the help of semialdehyde dehydrogenase to form alpha amino adipate. This alpha amino adipate converts to alpha keto adipate with the help of an enzyme amino transferase. This alpha keto adipate again undergoes dehydrogenation to form glutaryl-CoA. Glutaryl-CoA undergoes oxidative decarboxylation to form crotonyl-CoA, and this crotonyl-CoA finally converts to acetyl-CoA, which enters into ketogenic pathway. Coming to synthesis of carnitine, already discussed under beta oxidation of fatty acids. Here I am going to discuss only how it is synthesized from lysine. Carnitine is synthesized from ornithine and it is a 4-step process involving oxidation, splitting of glycine, dehydrogenation and finally oxidation. N-trimethyl-lysine undergoes oxidation with the help of trimethyl-lysine alpha-keto-glutarate dioxygenase, which is a dioxygenase enzyme which requires a molecular oxygen, finally converts to beta-hydroxy N-trimethyl-lysine. This beta-hydroxy N-trimethyl-lysine undergoes splitting to form glycine with the help of an enzyme serine hydroxymethyl transferase to form gamma butyrobutane aldehyde. This gamma butyrobutane aldehyde undergoes dehydrogenation with the help of NAD dependent enzyme dehydrogenase to form gamma butyrobutane. Finally, this gamma butyrine undergoes dehydrogenation with the help of an enzyme dioxygenase to form L-carnitine.